Again, today I'd like to talk to you about how we are testing quantitative adverse outcome pathway predictions using aromatase inhibitors within female fatty minerals. So toxicology is changing. Ever since the release of the report, Toxicity Testing Within the 21st Century, a Vision and a Strategy, there's been a push to make toxicity testing cheaper, faster, and more efficient. There are many ways that we are trying to accomplish this goal. One of those ways is to incorporate many advancements in other scientific fields, such as molecular biology and biotechnology, into the field of toxicology. Another way we are trying to do this is we are averting from whole organism testing, which is very costly and time consuming. Sometimes it can span multiple generations within the same species to test the effects of one chemical. And then when we do see negative effects at the end of these exposures, that gives us as scientists little mechanistic information of the changes that occur within the biology to cause those adverse effects. That makes it almost impossible to extrapolate between chemicals and the results of their exposures. So in order to better get a sense of these mechanistic changes, we have been focusing more on cellular-based testing. And in order to link cellular-based exposure results to whole organisms, adverse outcome pathways are being developed. Adverse outcome pathways are hypothetical constructs that attempt to link exposure data at lower levels of biological hierarchy to those higher levels. It does this by going through each level of hierarchy and trying to determine a key event that will occur there and going back and testing to see if it does. Adverse outcome pathways can be broken up into three basic parts, beginning with your molecular initiating event. This is the part at which whatever chemical you are studying begins to perturb the natural biological system of the organism. In this case, the molecular initiating event we'll be looking at today for our AOP is the inhibition of aromatase, CYP19, within the fish ovaries. Following the molecular initiating event are a series of key events. Key events are intermediate events that must be able to be experimentally quantified. Our AOP, our key events are the reduction of E2 synthesis, followed by a reduction of circulating E2, which causes a reduction of metallogenin production, which results in a reduction of circulating metallogenin, and finally leading to an impaired oocyte development. And our last part of our adverse outcome pathways are our adverse outcomes. They are the negative results due to the molecular initiating events that are of regulatory relevance at the individual or population level. Our adverse outcomes in our AOP today is the decreased ovulation and spawning within the female fish and a decline in their population. So adverse outcomes are great. Adverse outcome pathways are great qualitative constructs that can be used in hazard assessment. But to take that step further and get into risk assessment, we're developing quantitative adverse outcome pathways. Quantitative adverse outcome pathways aim to quantify the relationship between the dose of chemical used at the molecular initiating event to the probability and the severity of the adverse outcome that will result. It does this through a linking together of several interconnected quantitative models. The three models for our AOP that we'll be looking at today are the HPG axis model, the oocyte growth dynamics model, and the population dynamics model. This model encompasses several key events, as well as uses any results from previous models as inputs to the next model. So our goal is to demonstrate the use of this quantitative construct to simulate dose response and time course behaviors and predict the severity of key events along our adverse outcome pathway. Currently, this involves several key sources of uncertainty. Today, we'll be discussing two. First key source of uncertainty is species extrapolation from mammalian fish. Are we able to use mammalian-based in vitro data, plug them into our models, and create simulations that we can then use to predict fish outcomes? In order to tackle this key source of uncertainty, we will be selecting a subset of chemicals that have been shown to be aromatase inhibitors within a mammalian-based high-throughput screening assay, TOSCAT. We will then use those chemicals to determine their fish-specific AC50 values through a series of in vitro exposures to female fathead tissue. Now we will run our models using these data and compare the species-specific simulations against each other to see how much they are the same or how much they vary. Our second key source of uncertainty we will be looking at is whether it is important to use in vitro data versus in vivo data as our input parameters into our models. Short-term in vivo data will account for certain holistic processes that in vitro data doesn't, such as metabolism or specific chemical uptakes. And in order to tackle this, we are performing short-term in vivo exposures. And then we are using these results to drive additional predictions using our models. And then we will be comparing our in vitro-based simulations with those based on our short-term in vivo data. Okay, so how we got our chemicals. We looked at ToxCast. ToxCast is a mammalian-based high throughput screening assay. It has already screened over 2,000 chemicals, which is just a testament to the breadth these new age techniques can cover in such a short amount of time. Within ToxCast, we looked at 2 assays in particular to determine whether a chemical was an aromatase inhibitor. The first assay we looked at was a biochemical or cell-free assay, in which case the test compound was placed at the bottom of a well, followed by an enzyme solution and then a substrate solution. Our endpoint is fluorescence. If fluorescence is shown at the end of this assay, then our chemical did not inhibit aromatase. If no fluorescence is detected, then our chemical did sufficiently inhibit aromatase. The next assay within ToxCast we looked at was a cell-based assay. In this assay, the chemical in question was introduced into a cell, and if the chemical inhibited aromatase, it also blocked the transcription of luciferase. So the endpoint of this assay was luminescence. If luminescence was detected at the end of the assay, then the chemical did not inhibit aromatase. If no luminescence was detected, then the chemical did inhibit aromatase. So here is a list of the 8 chemicals we chose out of the 40 or so that were determined to be aromatase inhibitors through ToxCast. We chose 3 of these chemicals in particular: fadrazole, prochloraz, and propiconazole, because we have done previous 21-day reproductive exposures with them. We chose letrozole because letrozole is prevalent within the literature, and the other 4 chemicals are novel chemicals that we have not used to test them before. We chose these 8 specific chemicals because they range in a wide variety of potencies. We did not want to pick 8 of the strongest chemicals or 8 of the weakest chemicals. After we got our chemicals, we wanted to determine their fish-specific AC50 values. We did this by extracting ovaries from female fathead minnows, homogenizing the ovaries, and then adding radiolabeled androstenedione along with varying concentrations of our chemical. In its natural biological process, the aromatase CYP19 will convert androstenedione into estrone and water. And since the androstenedione introduced in the beginning of this assay was radiolabeled, the water will be tritiated that it produces. And tritiated water can be easily quantified using the fluid scintillation counter in the assay. So if the chemical does inhibit the aromatase, the amount of tritiated water we detect at the end of our assay will be substantially less than the amount in our control groups. Here's just a table depicting the average AC50 values for each chemical within each assay. Our 2 mammalian-based assays are on the left side, fish-specific on the right side. At first glance, it's easy to see that our fish-specific AC50 values tend to be a lot higher than our mammalian-based AC50 values. So in order to try to account for this and normalize the data, we decided to take the toxic equivalents for each chemical within each assay. Toxic equivalent is a way to weigh the potency of the less toxic chemical as a fraction of one of the more toxic chemicals. And our reference chemical we chose to make one is fadrazole. In order to calculate our toxic equivalent, we had to calculate our toxic equivalent factor. And that was easy enough. You take the reference chemical AC50 and divide it by your other chemicals AC50. We then plugged our toxic equivalents into the beginning of our first model, our HPG axis model, and developed several simulations. Here are just a few that I'm pointing out today. These are based on fadrazole. And so the first graph on your left is a simulation depicting what would happen in a 21-day exposure to circulating metallogenin concentrations at, when the fish is exposed to different concentrations of fadrazole. This is the final simulation we could obtain through our HPG axis model. We then took the results from that simulation and plugged it into our next model and developed the 2 graphs to the right. These 2 graphs to the right depict the decrease in ovulation and spawning. The box plots and the blue dots are the model predictions, and the green dots are the empirical lab findings that we found from our previous studies with fadrazole. As we can see, our model predictions are matching up with what we already knew to be true with the chemical. So that leads us to think that our model's on the right track. There are some additional simulations These simulations are based on a hypothetical 21-day exposure to 25 micrograms per liter of an aromatase inhibitor, and how that would affect the circulating E2 levels within the plasma. These three graphs represent the three assays we looked at earlier, the two mammalian-based assays on the left and the fish-specific assay on the right. Our days are on the x-axis and our E2 concentration on the y-axis, and the different colors represent the different chemicals that we were testing with. So when we compare these three graphs, we see that the overall profile of the graphs remain pretty consistent throughout the different assays, the mammalian and the fish. One thing that changes is the magnitude of the effect that is caused by the chemical, but it's still able to use mammalian-based, it appears to be able to at least use mammalian-based to predict some outcomes in fish. So our first key source of uncertainty is not as big a source of uncertainty as we first thought it might have been. So on to our next key source of uncertainty, whether it's better to use in vitro data or in vivo data to run our model predictions. We devised short-term in vivo exposures to generate the input data for our models. And in our short-term exposures, we exposed fish to 6 treatments of concentration. Well, we exposed them to one control with Lake Superior water and then 5 different concentrations of our aromatase inhibitor. 2 of them were below 25 micrograms per liter, 2 of them above 25 micrograms per liter. 25 micrograms per liter was our median because that was what our previous model predictions were based upon. So after the 24-hour exposure, organs of interest were extracted from the fish, and that includes their liver, gonads, their plasma, among others. And those organs of interest were then used to quantify our key events within our pathway. So here are some preliminary results for 2 of the chemicals that we were studying, letrozole and anastrozole. These results are actually based upon a 24-hour in vivo rangefinder study, which can be looked at like a pilot study. It has fewer concentrations of the chemical and fewer fish, but it does give us relevant information as to which concentration we should use that are below 25 micrograms per liter and which we should use that are above. So when we compare our lab findings at the bottom, the black and white graphs, to our model simulations at the top, the blue and white graphs, again, E2 concentration is on the y-axis and varying concentrations of our chemical is on the x-axis. And both of the model simulations are predicted for a 24-hour exposure. So we can see that there is a drastic reduction in circulating E2 within the first 24 hours, depending on the amount of chemical used within our model simulations. When we compare that with our lab findings, we do not see that. Our lab findings show that there is not a significant change in circulating E2 concentration, regardless of the amount of chemical used during the first 24 hours. We then did analytical chemistry on the plasma of these fish to see if we could detect the chemical within the plasma. No traces of letrozole were actually able to be detected within the fish who were exposed to letrozole, and only unquantifiable small amounts of anastrozole were found in the fish exposed to anastrozole. This has led us to hypothesize that perhaps these fish are rapidly metabolizing this chemical before they get a chance to affect their aromatase activity. This is the case in point and why it might be better to use the short-term in vivo data to run our model predictions, since it will account for such things as metabolism. So our next chemical we looked at for preliminary results, these are again based upon a 24-hour in vivo rangefinder study. We looked at imazolid. Here we are quantifying our first key event, the synthesis of E2, and we see that there is a drastic reduction in the production of E2 within the first 24 hours, which matches our first key event on our laparoscopic pathway. But then when we check to see our second key event again for E2 circulating E2 concentration, when we compare our lab findings to our model simulations, again our model simulations show a drastic decrease in circulating E2 within the first 24 hours, depending on the concentration of the chemical used, where our lab findings again do not show that. So this could be due to the fact that maybe there's so much E2 within the fish plasma that it didn't have time to dissipate by the time we chose to sample our fish. So this is an ongoing study, so some future directions that we're doing. We're definitely continuing to run our 24-hour in vivo exposures to generate the input parameters for additional model predictions. And then we will test both our in vivo based simulations and our in vitro-based simulations with a full-out 21-day reproductive exposure. Another direction we might want to take is to increase the length of our short-term 24-hour in vivo exposures, just in case we wanted to see if we could pick up on some of these later key events within our pathway. And finally, we want to determine chemical-specific differences in chemical uptake and elimination rates, and use those data as inputs to refine our predictive models. Again, as I said, this is an ongoing process, starting with model development, simulating those models, testing those simulations, and then going back to model refinement. I'd like to thank everybody here at the EPA, especially the bad mental team who helped me work on this, and our collaborators at the Research Triangle Park. I explained that really well, or I went too fast?
I really liked it. So how much more development does this need before we can use this in lieu of actual animal testing, like you said, more expensive, more time
Yeah.
consuming? So obviously be more efficient. What needs to happen before these kind of tests get implicated in actual?
We definitely need to address these key sources of uncertainty and make sure that we can't extrapolate between species and basically just make sure that we can make sure this stuff works right before we can change it. There's going to be a lot of additional after we do make our predictions. There's going to be a time where we are going to have to expose the animals to make sure that our predictions will work right. But I don't know, maybe 10 years.
Yeah, I mean, that's the whole purpose of that iterative approach, is to generate the simulations, test those predictions, and then keep refining until you get enough reduction in uncertainty that you're comfortable making decisions from that.
Okay, thank you.